We will now discuss expression summary methods. Expression summaries are the quantities used in all downstream analyses, such as differential expression, clustering, and developing predictive models. As a reminder, this is the general flow of a high-throughput genomic experiment. Once an array has been hybridized, there are various pre-processing steps, including image analysis for microarrays and alignment for RNA sequencing assays, which are then followed by normalization. We will now discuss expression summary methods. We will first discuss expression summaries for custom spotted arrays. For custom spotted array data, the quantity most frequently analyzed is the log base 2 ratio of the two channel intensities. Alternatively, this ratio may be calculated with background subtraction. Given that we may or may not apply background subtraction, I will call the numerator and denominator as sample and reference signal. You will need to ask those who ran the assay which dye, red or green, was applied to the sample and reference samples. The log base 2 transformation is applied because it facilitates interpretation and induces symmetry. That is, this transformation defines the relative abundance of a transcript in an experimental sample in comparison to the control or reference sample as the unit of analysis. For genes where the experimental intensity is greater than the control or reference intensity, the ratio could take values in the range from 1 to infinity. For genes where the experimental intensity is less than the control intensity, the ratio is compressed in the range from 0 to 1. The log base 2 transformation of ratio data promotes symmetry by treating under and overexpressed genes similarly. A typical example used to elucidate this concept is one that considers two genes, A and B, one with a ratio of 2 representing a doubling of intensity, and another with a ratio of 0.5 representing a halving of intensity. On a log base 2 scale, these values are symmetric about 0, indicating no change, with values of 1 and -1 respectively. The arithmetic average of these two ratios would be 2 plus 0.5 divided by 2, which equals 1.25. We note that the geometric mean would yield an appropriate measure of central tendency, giving 1. In fact, the geometric mean is the antilog of the average log ratios. Here we have added the log base 2 ratio to the table. The arithmetic mean is 0, and the antilog of this result is 1. Of course, we saw on the previous slide that the geometric mean, which again is the antilog of the average log ratios, is 1. The log scale tends to make the distribution more symmetrical. As an additional example, suppose we are interested in two genes, A and C, relative to a baseline gene, B. The absolute difference is markedly different for A and C, but compared to B, they both either increased or decreased by a factor of 10. Notice that on the log scale, the log base 2 of 100 is 2, the log base 2 of 1,000 is 3, and the log base 2 of 10,000 is 4, such that the differences are -1 and +1. We will now discuss expression summaries for Affymetrix GeneChips. Recall that an Affymetrix GeneChip is characterized as being composed of multiple 25 base sequences, each probing a specific gene or express sequence tag. There are 11 to 20 perfect match and corresponding mismatch probe pairs that interrogate for each gene. The set of PM and MM probe pairs that interrogate for a specific gene is called a probe set. To calculate gene expression, one needs a method of combining the data from the PM and MM probe pairs for the probe set into one overall summary. Typically, this is accomplished by applying some summary statistic to the intensity values for those probe pairs within a probe set. Again, the idea behind the mismatch probe is to assess nonspecific hybridization. So we would expect, or at least want, the PM intensities to be brighter than the MM intensities. As mentioned, there are roughly 11 to 20 perfect match and mismatch probe pairs that interrogate for each gene, called a probe set. The perfect match and mismatch intensities are combined to form an expression measure for the probe set. The early release of the Affymetrix MicroArray Suite software calculated signal as the average difference between the PM and MM probe pairs for each probe set after trimming the extreme observations. The average difference method presumed that the PM intensities represent specific binding plus some nonspecific binding, whereas the MM intensities represented only nonspecific binding. Thus, PM minus MM leaves us only with specific binding signal. Therefore, for each probe set, the average of the PM minus MM values was calculated. This average was taken to be the probe set expression summary. Formally, for each probe set, the average difference is given by this equation. Various researchers discovered problems with this probe set expression summary method. In fact, authors demonstrated that the PM minus MM difference was highly variable. This may result when the MM probe is actually measuring signal for another gene or express sequence tag. Also, when using the average difference method, when the MM intensities were greater than the corresponding PM intensities, a negative signal would result. Not only is this counterintuitive, but it made application of common transformations, such as the log, prohibitive because one cannot take the log of a negative number. To circumnavigate this problem, various researchers suggested that a constant value be assigned to all probe sets having an expression summary below some threshold. Problematically, when assigning constant expression values to some probe sets, this reduces the variance beyond what is real. Also, removing probe sets with negative expression summaries leads to the loss of information. To overcome the problem of negative probe intensity estimates, which occur when the MM signal exceeds the PM signal, which is outside of the physically possible range, starting with version 5.0, the Affymetrix MicroArray Suite software calculates signal intensities for each probe set using a new algorithm, which enforces positive probe pair differences. The MAS 5.0 probe set expression summaries are now calculated using the following procedure. First, background corrected PM and MM intensities are calculated by subtracting a weighted background intensity from each probe. Second, you subtract the stray signal as estimated from the ideal mismatch value. Remember, mismatch intensities are supposed to measure stray signal. However, if the mismatch intensity is larger than its corresponding perfect match intensity, we do not want to use it. Instead, we impute an appropriate value, and that is called an ideal mismatch value. Ideal mismatch values treat nonspecific binding, as estimated by the MM probe, as a proportion of the observed signal, rather than considering the MM intensity as an absolute signal reflective of nonspecific binding. Third, we log base 2 transform the PM minus IM values and then calculate Tukey's byweight estimator for the probe set. To estimate probe set signal, we then take the antilog of Tukey's byweight estimate and apply a scaling factor. Tukey's byweight estimate is simply a weighted average where the symmetric weights are defined by this equation. To this point, we have been concerned with problems of estimation in image analysis, normalization, and expression summaries. However, in most scientific activities, a great deal of attention is devoted to answering questions about the validity of theories or hypotheses concerning physical phenomenon, such as, is the gene present in the sample? The term hypothesis testing refers to the process of trying to decide, on the basis of experimental evidence, the veracity of such hypothesis. The output from the Affymetrix GeneChip operating software includes a column that indicates whether the probe set is present, marginally present, or absent in the hybridized sample. An example of the metrics tab data produced by the mass software appears in this table. These detection calls, P equals present, M equals marginally present, and A equals absent, produced by the mass software are based on a hypothesis test using the PM and MM intensities for the probe set. Rather than comparing paired PM and MM values directly, these detection calls are derived using the Wilcoxon signed rank test in the following manner. First, probe pairs that have saturated mismatch intensities are discarded. Additionally, probe pairs where the PM and MM are within tau of each other are discarded. If all probe pairs for the probe set are saturated, the probe set is automatically labeled present and the detection call p-value is set to 0. Thereafter, for each probe set, using each of its remaining probe pairs J, a discrimination score given by R is calculated. Then a Wilcoxon signed rank test is conducted for the probe set with the null hypothesis specified like this, while the alternative hypothesis is specified like this. This alternative hypothesis corresponds to testing that the gene is present. Note that tau is defined as some small non-negative number that can be altered by the user. The default is 0.015, which was empirically selected in a Latin square experiment. The decision rule is based on the resulting p-value. If the p-value is less than alpha sub 1, then the probe set is declared present. If the p-value is between alpha sub 1 and alpha sub 2, then the probe set is declared marginally present. And if the p-value is greater than alpha sub 2, the probe set is declared absent. The current defaults are alpha sub 1 equals 0.04 and alpha sub 2 equals 0.06. In both cases, the default values were selected to reduce the number of false detection calls. Many researchers examine the detection call field as a first method for filtering their data. That is, genes with a detection call of absent across all samples are removed from further examination. It has been observed that the variation of a specific probe within a probe set across multiple gene chips could be considerably smaller than the variance across the entire probe set. In fact, on the log-base 2 scale, the between array standard deviation is in general 5 times smaller than the within probe set standard deviation. This figure demonstrates this artifact for a specific probe set in a set of 4 technical replicate gene chips. That is, the same sample was hybridized to all 4 gene chips, and the probes vary more among themselves within the probe set than the probes vary between the 4 different samples. This figure demonstrates that same observation. In fact, among 16 technical replicates, which again means that the same sample was hybridized to all 16 gene chips, there was more variability observed between perfect match intensities within a probe set than between samples, and this variability is largest between perfect match probes than day that the gene chip was hybridized, module that the gene chip was hybridized in, or whether the sample was fresh or frozen. Therefore, to account for this strong probe affinity effect, Li and Wang proposed a multiplicative model where for each probe set on the kth array and for the jth probe pair for the probe set, the model parameters include V sub j, which is the mean intensity of the jth probe pair due to nonspecific hybridization. Alpha sub j is the rate of increase of hybridization due to the jth mismatch probe. Theta sub k is the expression index for the probe set on the kth gene chip. Phi sub j is the rate of increase corresponding to the specific hybridization of the jth perfect match probe, and E sub jk is the random error term assumed to be Gaussian or normally distributed. The model assumes the probe intensity will increase as theta sub k increases, but the rate of increase is different for the different probe pairs within a probe set and also differs for perfect match and mismatch probes. Using these two equations, the probe pair difference is modeled by this equation. There are then n by j equations available to estimate n plus j parameters. The estimation procedure is an iterative process whereby first the phis are assumed known and are used to solve for the thetas. Once the estimates for thetas have been obtained, they are subsequently used to solve for the phis. This process is repeated iteratively. For analysis where multiple arrays are available, a model-based expression index is defined as the least squares estimate of theta hat sub k of the expression parameters. So in this model, theta sub k is the expression index for the probe set. In order to solve this problem numerically, a constraint is applied to ensure identifiability. The software package DChip can be used to fit this model and obtain model-based expression index expression summaries. This software also applies invariant set normalization prior to calculating the MBEI expression summaries. Some researchers propose that expression summaries should be based only on the perfect match intensities. In fact, this idea is generally applied in the Robust MultiArray Average method, or RMA, which is the most commonly applied probe set expression summary for Affymetrix GeneChips. The steps for obtaining RMA expression summaries are to first calculate background and adjust the perfect match intensities for background. Then apply quantile normalization to the background adjusted perfect match intensities, log base 2 transform the results, and then fit a median polish model to each probe set. The median polish is a robust estimation procedure and is used to estimate the model parameters in order to protect against outlier probes. After the median polish, the estimated parameters are interpreted as follows. Mu hat is the overall log base 2 probe set effect. Alpha sub k is the log base 2 effect of the kth gene chip. Beta sub j is the log base 2 effect for the probe. And E are the error terms. Then the probe set expression for gene chip k is given by mu hat plus alpha sub k hat. As a reminder from the normalization module, RNA sequencing data are usually expressed as reads per million, reads per kilobase per million, fragments per kilobase per million, or transcripts per million. When analyzing these, they are log base 2 transformed. Note that raw counts can still be used in downstream analyses when you use selected analytical methods. Here are some related references.